Today's presentation is COVID-19 Outcomes Database, Insights and Opportunities. Our speaker today is Dr. Jaap Moendema. Jaap is currently the Chief Innovation Officer at Certara. Earlier in his career, he was a pioneer of model-based meta-analysis. Jaap's research interests include applying modeling and simulation to optimize treatment strategies, trial designs, and decision-making. Jaap, welcome to the event. I'll now turn it over to him to begin the presentation.
Thank you. Excuse me. Thank you very much for the introduction and welcome to everybody. I hope to give you an overview today on the outcomes database that we've built around COVID-19 and some of the applications that we've derived from looking at the data that's collected inside that database. And take you back to some of the objectives when we started out this initial project sometime in April, end of April last year, 2020, where our objective was really to curate and augment all publicly available aggregate data on studies evaluating pharmacological interventions, risk factors, or biomarkers for COVID-19 disease progression, treatment, and prophylaxis. Quite a substantial undertaking, and we've done some of those, and I'll show you in detail more of what actually we got to. We wanted to make this data freely available for research purposes. We have made this data freely available for research purposes, as it was funded by the Bill and Melinda Gates Foundation. And we wanted to facilitate access to that particular data by providing a web-based interface, or CODEX platform that was wrapped around the database so that you can quickly summarize, zoom into parts of the data, make graphs, and do some automated analyses. But also to facilitate access, we are a partner of the International COVID-19 Data Alliance, or ICODA, which is a platform in which pharmaceutical companies as well as academic researchers make data from clinical trials available for particular research purposes. And the data accumulated in our COVID-19 database is in there as well. So why did we collect this data? Well, one of the key applications that we wanted to look at is, as new information comes out during the pandemic, to be able to quickly interpret those outcomes in light of existing evidence on things that already have been evaluated, to make some judgment on the value of particular new treatments that are coming out, both from randomized controlled clinical trials. I wanted to see how well we could use observational studies, or maybe even data from single cohorts. Also interested in quantifying risk factors, both for absolute and relative risk for a certain level of disease severity, mortality, and duration of hospitalization. And really, COVID-19 is a complex disease that changes over time. To understand that longitudinal relationship between the viral load, vital signs, biomarkers, and outcomes. All to understand the importance of the time of treatment initiation on the course of the disease. So what did we pull together? We pulled together all information on studies that evaluated treatments, as long as it had more than 10 patients in it. And these were randomized controlled studies, as well as retrospective cohort, observational cohort studies, but mostly controlled. So initially, we also looked at individual cohorts, but decided later to minimize that information. Really looking at any treatment, any type of drug that has been evaluated. So there's pharmacological interventions, collecting all patient characteristics of the patients in those studies, as well as any outcome, looking at symptoms, viral load, biomarkers, and more the clinical efficacy and safety outcomes as well. On the risk factor side, we included studies with cohorts larger than 200 patients that really evaluate patient characteristics stratified by outcomes. So that you can look, okay, if we take male versus female, is there a higher risk of dying once you get hospitalized? Similarly for particular comorbidities, as well as other aspects that are out there. Maybe potentially to use that to adjust for differences in patient populations in our analyses. And then the third factor is the CODEX platform, as I mentioned, to provide online access to it. And we expanded that quite a bit to include automated analysis on that data. So you can do automated pairwise meta-analysis as well as network meta-analysis for the risk factors and the interventions that have been evaluated. Our data comes from a variety of sources. We use the NIH indexed COVID as our main source of publications, and serves the preprint servers that are there, as well as track on the registries all information and studies that are being registered there. And as soon as information comes out in those platforms.
Yeah. Sorry to interrupt you. Can you make sure you're speaking into the microphone, please? We're having a little bit of a hard time hearing you.
Okay, I'm sorry about that. Is that
Yep,
better?
that's better. Yeah, that's better. Thank you.
Okay, I'll hope to to be able to do that. I'm used to walking around when I give presentations, which is much harder when we do this remotely. So the information, where does it come from, and how do we review that? So we've collected about 100,000 sources of information, so quite a few. So we have to index that and provide an algorithm to identify the particular treatments that are being evaluated, what type of study it is, what type of evaluation that's being done, so that we can flag a subset of those sources for primary review. And about a little over 5,000 are flagged for primary review, and still mostly out of the published literature. And those are being reviewed, and this graph actually shows the flow of information. We did our first search in May, on 5th of last year, and since that was the first one, it had a little bit more, but then there's a steady flow of about 10,000 publications coming out on COVID-19, of which about 5% contain information so that evaluate particular treatments, and those are the ones we initially look at. And I've reviewed of the 10,000 actually that we have reviewed right now, about 7% have been flagged for inclusion. So it's about 750 sources that provide information on things that we're interested in. And that's still quite a bit. So we came up with a prioritization system to give priority to randomized controlled studies. And so far, all the randomized controlled studies that have reported outcomes are, as far as we know, included in our database. And then it's augmented with larger observational studies. So if we focus back on the treatment, which was one of our main aspects that we were looking at, understanding the impact of particular pharmacological interventions on the outcome for COVID-19, about 440 flagged, of which 264 are into the database. Going from there, they start to, once we flag those indications, our team in India extracts the information from those sources and builds up our initial database, which is then further augmented and structured by our team in Menlo Park, providing additional information, because we have to understand and put some commonality into definitions that may vary quite a bit from study to study, especially on the severity of the disease of the patients, as well as calculating a lot of the things that are not being published, but that we can calculate from the information that's available. So that's all done, put together, and then provided access through a web-based interface. And if you would pop that up, if you would fire up our application as it sits online, and maybe some of you have been able to do that, you'll see this landing page where you get in, where you can get an overview of what's actually currently inside the database. And it's, like I said, information from 282 references containing information about 295 studies, representing a little bit over 325,000 patients in this particular database. So it's a large source of information. Death is the most commonly reported outcome, actually presented in most of the studies. And then there's a big drop-off. Only about 150 studies focus on hospitalizations, patients progressing through critical disease, or other outcomes. And it starts to go below 100 once we've looked at the top 5 outcomes from the efficacy perspective. Tocilizumab is actually the most studied intervention, reported in 53 studies, followed by hydroxychloroquine. We'll focus on these two quite a bit in the rest of the presentation. Convalescent plasma after that, and antivirals, lopinavir, ritonavir, and hydroxychloroquine combinations after that. And actually, there's probably about, I don't know, quite a few interventions that have been evaluated, probably going over 80 total. So a lot of them only in one or two studies. But 34% of the information comes from randomized controlled clinical trials. So in this particular application, you can filter and zoom in to a variety of the data, the left-hand side, and do a variety of things with the data on the top. And like I mentioned, one of the aspects that we can do is we can actually do an automated network meta-analysis of the randomized controlled trials. And the picture may show up, as you can see here. It looks at all the randomized controlled studies and how they are connected to each other. Most of them are compared to standard of care, so that's actually our big point here in the center. And all these particular treatments, you may not be able to read them all. There's quite a few are given on top of standard of care compared to the particular standard of care, and a few have active comparisons in them. But most of them, it's a simple comparison of standard of care versus the treatment plus standard of care. So let's look a little bit more into when and how this information is coming out. Obviously, during a pandemic, we want to react quickly. And as information comes out, we want to be able to aggregate that and understand the implication of it. And if we look from the start of pandemic months, we start in January of 2020, of the information that's coming out over time, of the studies that sit in our database. Initially, most of them are observational studies, with a few in the second pandemic month, we get one or two randomized controlled studies, and getting a little larger majority as we come along. So a large fraction of this information that we achieve initially comes from observational studies. And then the fraction of randomized controlled trials is increasing. We see a drop after month 5 because we still need to get a lot of studies reporting. This is the start date of the studies, so studies that started later in the pandemic still haven't all reported their outcomes yet, and we're waiting from them. You may see quite a bit larger fraction here of randomized controlled studies. That's because we prioritize them going forward as being included in the database. So a question comes out, or a question we may ask ourselves, since we get so much information from these coming out from these observational studies, what can we do with that? Before we look into that, here's an overview of the information now clustered by treatment groups. So each of the individual treatments are being tracked, but I've lumped them for presentation. And as I go through the presentation, you may see some of these groupings to make it a little bit easier to understand the outcomes. Mostly studied immune modulators. I'm putting the IL-6 and IL-1 compounds into here, such as tocilizumab and anakinra, hydroxychloroquine, a large number of studies, randomized controlled as well as observational. And actually, if we look at the total pool of observational studies, there's quite a few more smaller ones out there. Corticosteroids, antivirals nicely balanced between those two sources of information. So let's dive into this a little bit. Let's look at some actual outcomes of the information. And here I have a first slide that zooms into that. It's quite a busy slide, so bear with me. On the top of the slide, you see the outcome for mortality. So we look at the risk ratio here for death. Meaning, if you're on the right-hand side of the line of 1, that's a bad outcome. If you're on the left-hand side of 1, that's a good outcome. And there's 5 randomized controlled studies about tocilizumab, which I picked as an example in this particular slide. Although the trends I'll talk about are consistent across all the other treatments. We see these studies seem to be quite consistent with respect to their outcomes, all close to 1 or slightly above 1, and on average, there's a risk ratio of about 1.1. So not providing a particular benefit for mortality in these randomized controlled studies. The reason I looked at mortality first is because that's an endpoint, especially if you have a lot of smaller studies where aggregating data makes sense to get better insight, as well as obviously mortality is still quite high, so it's a very important aspect of the disease to be able to modify. Now, if we look at the observational studies that are listed out here, we see something that looks a little differently. We see quite a bit of variation in the outcomes that is reported in these studies. And actually, that's quantified in a way. We can look at the variability in these studies beyond what we would expect just purely on sample size alone, because obviously, if you have smaller studies, the responses will vary more. Once and it should tighten up, but you get more larger studies that are listed on the bottom here. But even once you correct for the sample sizes, there's quite a bit of additional heterogeneity in the treatment effect that's being measured in these observational studies, and that's quantified by this particular parameter, tau squared. So that's one aspect. The other aspect that we see is they all lie to the left-hand side. Actually, they all seem to present a much more positive outcome for tocilizumab than if we take randomized controlled studies as our particular gold standard. You would see from there, and on aggregate, that difference is quite large, 0.6 for the risk ratio, so about 40% risk reduction on aggregate in these studies that report the observational studies that report death as an outcome. And this is a general trend. We actually see that, as I'll show in more detail, in all of these studies. So let's dive into that a little bit further. Obviously, the observational studies try to do something to correct for a particular issue that they have. We look back and compare patients that have received a certain treatment to a cohort that has not. It's not randomized, so those cohorts may differ quite a bit. And they apply certain methodologies in those studies to correct for those differences. For example, they may apply certain algorithms to try to match the patients that are receiving the intervention versus patients that have not received a particular intervention. And those are shown on the top here, where a particular matching algorithm was being used to evaluate mortality outcomes. But that doesn't seem to get rid of the bias that we observed on aggregate. These studies still report a similar 0.6 on aggregate reduction in or for the risk ratio. So would suggest quite a substantial benefit of this particular treatment with significant heterogeneity. If we look at another methodology to try to account for patients ending up in these cohorts that are being evaluated, doing a multivariate analysis to account for the patient characteristics that could be different in these studies, that doesn't seem to be addressing the issue either. If anything, it makes it worse. And then there's a set of studies that actually don't do anything to account for the differences. For some of the smaller studies, that's expected because it's hard to do that. But even the larger studies that are listed here, it meant that mortality actually was not the primary outcome in these studies. They looked at another aspect for which they did a multivariate analysis to account for the outcomes. So that's one complexity with the observational studies that we have as well, is that the focus is often a particular outcome. And it may not be the one that we're interested in and looking across all of these studies. So whatever was done, either matching or a multivariate regression model inside, so within the study comparison, seemed to address this particular bias. And if we look now, replicate this across all the different treatments that are there, we see that pattern occur. And bear with me, this is now quite a busy slide. we're still looking at the risk ratio for mortality. So 1 means no benefit. Our big blue squares are the estimated treatment effect for clusters of treatments. Let's zoom in back here on our immune modulators. We had our randomized controlled studies estimate an effect. We had our observational studies in green that did some adjustment or tried to account for the patient differences, or in black where nothing was done. It's just a raw mortality outcome that was being reported. And we see in general across all of these particular treatments that have been evaluated, in certain instances more than others. For antivirals and immune modulators, it's quite pronounced. They would suggest a much better outcome that we would anticipate from these treatments than what is observed in the randomized controlled studies. So obviously from this perspective, if we would take randomized controlled studies as our gold standard, which I think most of us would, looking at these observational studies can be problematic, as they all seem to provide a much more optimistic view, maybe apart from the antibiotics here on the bottom, an optimistic view of what's going on. The additional heterogeneity is consistent as well. So apart from that bias, we see much more variability in the outcomes, but that's something we can overcome. We can, just means that you have to do more of them and look at repeated studies and get rid of that variability by taking the average across a few studies. The bias is a little bit harder to address. But we try to see whether we can use that wealth of information that we have in our database to really account for these differences. For example, if we zoom into another part of the application, looking at risk factors, here you see the impact of patients having diabetes. For patients not having diabetes on the outcome of mortality, again, on average, there's quite a significant increase in your odds of dying from COVID-19 once you're hospitalized. It's about 1.7 on the odds ratio, and all of these studies show that consistently. And there's a variety of these factors. So we're thinking, can we combine that and build up a model to account for all these patient differences? Before I do that, I want to go back actually for those of you who are not as familiar with the disease, and think a little bit about why that is so important. And the reason of that is twofold. One is we have a very large variation of the patients that show up in these studies with respect to their severity of the disease, which can range from patients with mild to moderate illness, meaning they have symptoms or they have some sign of lower respiratory tract infection, up to quite severe illness where patients are on oxygen or receive invasive ventilation. So this large variation obviously can interact with the treatments as well as interact with the patient journeys. And some of them will progress from these early stations over time into more severe illness, where with respect to disease, something is happening. Initially, it's the viral replication that is the most important representation. But after that, it changes to more of a host disease where the patient's response to that with respect to their immune system becomes the driving force. And the therapies that are being evaluated obviously change over time, where the immune modulators or the anti-inflammatory therapy mentioned here, more later stage, and antiviral therapy more early stage. So that complex time course of disease and these large variation in the severity in the patients that end up in the studies is a very important factor to account for. And most of the studies report some information on that, but not all. And this is one of the complexities, obviously, that we have to deal with when we look at publicly available data. And one of the learnings, and also another mention I want to make on the ICODA initiative, where more information on especially some of these outcomes is actually being shared on the trials, much more than what's being published, because we need that to understand that full interaction. sex, age, and disease severity group we can identify for all of these studies. And actually, on average, 20% of these studies report the information of the outcome stratified by either male, female, different age groups, or different disease severity groups. So that's quite an important aspect of the data. So we'd like to use that. We'd like to use those within study comparisons to really enhance our understanding of these risk factors. But a very important one, such as disease duration, only mentioned in about 50% of the studies. So there's quite a bit of information lacking there. And if we want to use that, we want to dive into, we have to impute and build up a model to fill in the missing pieces on these attributes of the patients that end up in the studies. And we do that through the correlation. Actually, no surprise. So some of these are highly correlated. When patients get older, they have more hypertension, they have more diabetes, they have more other aspects. So from some of these relationships, as you see here, presented age versus hypertension, we can learn and build up an understanding of what most likely the patients would have been in each of these studies. And again, no surprise, disease severity, as we've classified them in critical going to mild, is one of the most important differentiating factor and highly correlated with some of the other outcomes. The typical patients is older in critical disease versus mild moderate disease. It contains more males, more patients with diabetes, more patients with hypertension, more patients that are obese, and obviously in these high categories, the majority is on oxygen support and have had the disease for a longer period of time. So we built this information into our particular analysis and try to see whether we could use that to account for what we saw, the bias we saw in the observational studies. And unfortunately, that did not work. So we built a nice multivariate model. We understand very well how the patient's factors impact these outcomes. And I'll show more in detail on that. Age, disease severity, being male or female, or diabetes were highly significant on the outcomes or the odds of dying due to COVID-19, hypertension, disease duration, whether the studies were done in Asia or mostly China or not in China, or obesity did not impact these outcomes. And some of those may be surprising. Hypertension by itself is an important factor, but once we put diabetes in and they're highly correlated, it disappears. And obesity is not an important factor either by itself or in a multivariate setting. Again, blue is our randomized controlled studies that we looked at. In green, we have our observational studies that we try to adjust with this model. And we may have moved the needle a little bit, but could not get it to where our randomized controlled studies provide insights. But importantly, if we look at our randomized controlled studies, some of them present information of treatments that do seem to work. In this particular instance, our corticosteroid group actually on average gives about 0.83 odds ratio for dying, and it's highly significant benefit just from those studies alone. Antiviral therapy on average, no impact. Antibiotics on average, no impact. And actually hydroxychloroquine, we can quite certainly say that definitely no benefit. It could actually provide harm to the patients to which it's given, as we are above the line of unity here with the information coming out, about 10% increase, 1.1 odds ratio of dying on the patients that actually receive that particular treatment. So it means that we're stuck with our randomized controlled studies to really get the insights on the different treatments. So that's a slide you see here. I'm still focusing on mortality as the outcome. I picked the first 25 treatments, so instead of clustering, now we can look at the individual treatments and see how they impact patients dying due to disease or surviving. And they're sorted by the precision. So where we have most information show up on the bottom here. Our lopinavir, ritonavir, very much studied antiviral combination, no benefit. Like I mentioned before, hydroxychloroquine actually could be causing harm. Our corticosteroids providing benefit, dexamethasone shows up significantly, and some of the other ones we see a trend. Interferons not. Remdesivir may have a particular a trend to provide some benefit, not clear. Tocilizumab as our IL-6 does not from a mortality outcome, and maybe convalescent plasma has some opportunity there. So one of the things to look at, again, from now a pandemic perspective, what you saw in the previous slide is the insights we have now, being in January of 2021. Obviously, we would like to look at this, and here I provide a timeline of information coming out on hydroxychloroquine. I pick that as my example. So we see all the studies listed here, going from April 2020 all the way to January 2021. And on the right-hand side in the graph, we see our average estimate as information flows over time coming into this particular database, and how our understanding of the impact of this particular treatment on mortality changes. And initially, really, there was no reason to believe that this particular treatment could have provided any benefit focused on the mortality outcome. I still included both observational studies, despite the potential bias, as well as randomized controlled trials in this study. And that's purely because we have to do something with the information that's coming out. But I'm down-weighting the observational studies by accounting for the heterogeneity that they would provide. Obviously, initially, we don't know that, but we could build it in as a fixed component once I'm analyzing the data as it goes along, and I'm discounting that, assuming a certain variability. But anything, there is a big study that happens in about the May time frame that may have pulled this information down. We're up to about 1,000 patients yet. Still, no real huge optimism that would present this would work very, very well. And that's around the time that the FDA granted the emergency use authorization, which, as soon as the first large randomized controlled study came out, the recovery hydroxychloroquine study out of the UK, where it added about 4,000 patients. And there was a randomized controlled study, this early one was one as well. But at that point in time, we get a very clear insight that really this particular treatment does not provide any benefit. If anything, it could do some harm. Yet, even beyond that point, there were another 50,000 and probably quite 40,000, probably quite a few more patients that were exposed in particular studies to this treatment. And so quite a few of these studies started later compared to this particular aspect. So we have to ask ourselves, can we use on a global scale the information that we need to quickly respond to it much better, and not have that being used on a treatment that actually very early on we would have enough insight in did not provide any benefit. The FDA luckily acted quite quickly, where they revoked the emergency use authorization somewhere around the end of June time frame, responding to the information probably that came out of the recovery study. But we have to ask ourselves whether it should have even been granted early on in the pandemic. So let's look a little bit more at other aspects after gone on looking at that particular treatment that did not work. We have now a very good understanding of in these studies what are the aspects of the patient populations that impact the outcome. So I mentioned it earlier, we build up that large model, understanding the impact of all of these factors on the outcome. And still age, no surprise there, is one of the most dominant factors. For every 10 years, there's about 1.6 odds ratio of dying. Diabetes, same, as about 10 years of age difference. And being male versus female is a disadvantage as well. Obviously, the disease severity is a very important factor, where if we take moderate disease, moderate means patients are receiving oxygen as our reference, that patients who are severe, meaning they may need mechanical ventilation or high flow oxygen, quite a big increase, and about threefold increase in odds if patients start entering the study in the critical disease level. And we've built that information on it over time as well. And you know, within about 5 weeks, we've achieved of most of the patients that will not survive in these particular studies will do so by about 5 weeks. Hypertension, obesity, also the standard of care. We looked at it quite significantly. Standard of care may vary from study to study. Of course, all these treatments are compared standard of care on top of treatment on top of standard of care. But what the particular standard of care was in these studies did not seem to impact the outcome much. Neither did time since pandemic start. And that was an interesting feature because we would think that maybe we've gone a little bit better over time in addressing the disease. But at least the patients that are reflective in the studies that are being reported, there is no change in average mortality as we went from the 1st to the 6th month of the pandemic. Visually, we can look at this here. Here you see a picture of over time, and this is now time since start of intervention, of what happens with the patients, the proportion of patients that will be dying, depending on whether they have mild, moderate disease as well or critical disease. And once you get to the stage of critical disease, the picture is quite bleak, where if we look at a typical patient population, slightly under 40% of the patients will not survive. But obviously there's some correlation. These patients are also older, have more males. So once we take that into consideration, once you're at this stage, there's still 50% chance of actually not surviving that treatment. So it's very important that we keep on looking at interventions that could help and improve that outcome, or help prevent patients getting to that stage of critical disease. You see 2 areas in here. The dark blue area is our confidence interval and how well think we know that particular trajectory. And the light blue area is a prediction interval, so what we expect the next study to show. And the reason I put that in is because it's still quite large. There's a lot of variability. We've explained quite a bit of it by our model. Actually, about 40% of the variability is explained by the model. But we cannot even use this to serve as an historic control for single cohorts. There's still so much variability left. It's not worth a whole lot of patients. So it's more important to understand the patient journey and what particular patients that you have in your study will drive the outcome. Similar graph for age. I'm going to skip through that quickly due to time and look at pandemic month, where at least up to month 6, we have not seen much improvement. So now let's look at another aspect of the patients. Of course, there can be an interaction between the type of patients that we get and the particular impact of the treatment. So if we look and I cluster the treatments a little bit more with respect to their severity, so severe category here is both critical and severe. Moderate is still the same, and I have mild, which also includes the asymptomatic and other parts. We can start to think we have quite a bit of information across all of these different patients group for these clusters of treatments. We can look at the interaction of that between these levels of disease severity and the outcome of a particular treatment. And again, here I wish that every study would have reported the outcome stratified by disease severity. Only about 20% of the studies do so. On other aspects where information accumulated in the ICODA initiative could be beneficial and better understand these outcomes. A very important one, if we do this, shows up on our corticosteroids. Remember, that was one of the treatments where we've clearly shown a benefit of treating the patients with outcome, but that benefit is mostly there in either the severe patients, actually that's where it's largest, the red dot in this circled area for corticosteroids. The severe patients show about an odds ratio of 0.66, moderate patients 0.8, so not as effective in that, but still significantly. So for any patients that have achieved this inflammatory stage of disease where they are required oxygen, we see that particular benefit. But actually the trend, there's not as much information there because the steroids aren't luckily given into those situations a lot, but where the patients are milder, that may actually flip around and it may be harmful to administer these particular treatments. So very important understanding this interaction between treatment and some of the aspects of the disease severity. Unfortunately, it did not improve. Let's focus on our antivirals. That would be one where we may anticipate that our patients are earlier in the disease, where they still have a mild disease, that would enhance the outcome because there's still a lot of buildup of virus going on. But we do not see across all of these stages, the average estimate is close to 1. No clear benefit there in either. And in all of these particular areas of the disease, similar hydroxychloroquine is doing potentially more harm than benefit. We can look at some other interactions. AIDS, I'm going to skip that. Look at the time since start of treatment. That's another one. How long did patients have the disease? And I circled our antivirals here because that's where we think that may have a big impact on the outcome. And if we look at patients where they typically had the disease since first onset of symptoms for less than 7 days, not a clear benefit or difference from the typical where they have the disease for more than 7 days. So a similar no effect of all the antiviral therapies that are out there. So of course this focuses on mortality. Obviously, still a large fraction of the patients survive, and what important to know is what happens to them. How severe a disease do they have? How quickly can they leave the hospital? And haven't fully built that up yet, but wanted to show some information on hospitalization. So how we analyzed the mortality data, we did something similar on the hospitalization side, and we understand, no surprise, similar factors that drive the odds of not surviving due to COVID also drive the hospitalization. So I presented it here on the same scale. Even though we analyzed hospitalization discharge, I'm looking at the hospitalization here, and it's the similar factors that drive it. The big change is critical disease is actually now a much larger difference. It really delays the, for obvious reasons, patients stay in the hospital much longer than the other treatments. So actually not being able to modify how the patients end up in each of these categories could be a clear benefit. And we can put those two together. What I've done in the slide here is present the similar picture we had before in blue, the mortality over time for each of the disease severities, and now look at the hospitalization over time coming down, where at around 35 days, these mild and moderate diseases, these lines start to meet each other. It means the patients are either discharged or they have not survived. Whereas in the critical disease, and obviously that time period is much longer and not even not close together yet. So there's still a fraction of patients that are being hospitalized in this remaining area. So it warrants to look at that. Actually, let's look at the outcome of hospitalization in the last few minutes I have before I wrap it up, because we could put those together. So what I've tried to do in this particular slide, showing the impact. Now, again, this is all randomized controlled studies to minimize the risk of bias, look at individual treatments, compare hospitalization, as well as mortality. Where on the left-hand side of one, we have a good outcome, mortality in red, hospitalization in blue. And for our corticosteroids, such as dexamethasone, that's listed, we see a benefit on both the hospitalizations and mortality. But we see something interesting occur from our remdesivir information, as well as our tocilizumab as an immune modulator, where there may be some benefit on the hospitalization side. Patients seem to either not progress as far in the disease on the treatment and be able to leave the hospital sooner on remdesivir and tocilizumab. Yet there's very little impact on the mortality outcome. So that really warrants the question, why is that? Why do we see this sort of separating effect on one aspect of the disease, but not in overall mortality? Another interesting aspect is to look back at our timeline of information coming out. Of course, death is a pretty hard endpoint. Hospitalization may have more information on that, a little higher precision. And if we look at in blue on that timeline, again, very early on, no reason to believe there should have been any benefit of this particular treatment. So really, what's next? In the next stage, and thank you very much for your attention on this, I really hope to be able to split up the disease into these stages further and understand the interaction better between those levels of disease severity and the particular outcomes as they relate to treatments. We've looked at the both ends, hospitalization and mortality. There's a lot in between that we need to better understand and could provide quite some benefit of treatments that modify how patients progress through these particular stages. So that's where I wanted to leave it. I thank everybody very much for the attention, and open it up for questions.
Fantastic. Thank you so much, Jaap. Got a couple questions. We'd love for our attendees to enter their questions for Jaap into the questions panel. So someone just wanted to make a comment that they felt that antibody therapy could also be used for prophylaxis. I didn't know if you wanted to comment on that, Jaap.
Yes, antibiotic therapy is pretty much standard use in the treatment in the hospitalized patients, especially if we going to get to more severe disease. So definitely that's used here. We're looking mostly at azithromycin as a particular one that had very early focus.
Super. Someone was hoping that you could elaborate, please, on how you weighed randomized control trials versus observational trials.
That weighing was done by accounting for different levels of heterogeneity between those studies. So we assume there's no bias, and it's just a level of variation. On average, the results could present a similar outcome, and allowing for that additional heterogeneity will prevent a different weighing between those two type of studies. So that was built in. So we have a different level of heterogeneity. It's actually close to zero for the randomized controlled studies for any of the treatments, and on average, and it's still quite substantial for the observational studies.
Okay, great, thank you. Looking to see if we have any more questions from our audience. I think that may be it. I'll just pause for a second. All right, I think that that is all the questions from our audience. So someone wanted to know, I've got a couple more. How do you capture and quantify your heterogeneity?
That is measured, or that's estimated from the additional between trial variability that's there in the treatment effect that's measured in each of the studies. So there's two levels of variation, and I did not elaborate enough on that while I was presenting. One is just the absolute variation in outcome, and that was addressed with the covariate model that I showed, how aspects impact the overall variation in response. But then there is the within study estimated impact, either on treatment or a risk factor, and how that varies from study to study, because some treatments are duplicated across studies. So that aspect is estimated from the duplication of particular interventions or group clusters of interventions across studies.
Thank you. Someone wants to know, I think I understand this question, but we may need clarification. They say that the impact of some of the discussed covariates is compound. For example, diabetes, treated or untreated, or obesity. So I think what they mean is some folks that have diabetes also might be obese. In order to know the role of a single factor, you will need to substratify the patients, which might not be statistically reliable. How did you deal with that?
Yes, so we deal with that in two ways. One is when I showed the multivariate model, and thanks for the question, because I forgot to address that when I showed the slides. When we looked at the multivariate model to understand the patient characteristics, I included any stratification that was reported in studies for anything that could be potentially of interest. Obviously, a within study comparison is much more powerful to estimate the impact of these variables than in a cross-study comparison. So if studies report stratified results, I include all of those and weigh them appropriately so that each of the studies still contributes only one overall. And we've done it in oncology applications as well to try to understand variability in patients in cancer studies, as well as other areas. So that's one aspect, but obviously there's correlations. I do not have all these multiple stratified outcomes. That would be ideal to have it subsetted in all of these different categories. We account for that a little bit because we know that correlation. We know that patients that have diabetes or not have diabetes are all older, and that's accounted for because the age is reported in certain aspects in these patients groups. So some of that correlation is built in that aspect. So that's one way how we address that. And when I showed the interaction between treatment and outcome, those are actually based purely on a single representation of the study, where we zoom in on the stratification that's being reported. So when I look at disease severity, I only isolate studies where I know the disease severity group, or studies that have stratified information by disease severity, and analyze that aspect. When we look at age, we do that again. So actually, it's not one analysis I presented. It's probably about 15 different analyses that I looked at across these different slides that are presented. It's not one analysis to address all of these aspects, a variety of different analyses to do so. So thanks for that question, so I could elaborate on that a little bit.
Super. Someone wants to know if there were any studies with a direct comparison of azithromycin versus lopinavir. If so, which was more effective?
Unfortunately, I do not know that on the top of my head, so we would have to look into that. I'm not set up. We could pop up the CODEX application and have it live at this point in time. And look at that very quickly. You would see whether that has been evaluated. If you pop up the network plot of all the studies that are there, and then you could look at that particular study, or from a network perspective, try to understand that comparison. But I would not know that on the top of my head.
One of our attendees wants to know how you handled sample size variation from study to study.
Well, what's used in the analysis is the, let's say we focus on mortality, we use the proportion of patients that would die as the outcome, and the standard error of that proportion as the factor that's included in the meta-analysis to weigh the different studies. So the sample size comes into that standard error, or the precision of the estimate out of each of the studies.
Looks like we got a few more questions. Someone wants to know, what software did you use for the multivariate statistical analysis, and what are the most important variables?
Everything, all the meta-analysis were done with Metaphor. The particular multivariate meta-analysis regression tool that's available there in R. So that was the tool that was being used. You can specify multiple levels of heterogeneity, as well as multiple potential factors that impact either treatment or overall response. So that was the particular analysis tool that was being used, Metaphor in R for any of the analysis, actually all of the analysis that I've presented here.
Looks like we've got just a few more questions, and I think these are kind of maybe can get answered at the same time. One person wanted to know if you've created regression mathematical equations from your model, and then someone wanted to know if you used meta regression techniques to investigate the effects of multiple factors.
Yes, and yes. So multiple factors are considered all at once in a multivariate setting, as I explained before. So they're all regression-based meta-analysis techniques.
Okay, great. Thank you so much, Jaap, and thank you so much to our audience for all those excellent questions. Jaap, if you can go to the next slide, please. So we'd like to invite everybody on the call to our next webinar in the series. It will be on February 3rd, where Joanne Malatesta and Daryl Nix will present Enhance Your Drug Development Capacity with ClinPharm Study Execution and Operational Support. And then lastly, on February 18th, Patrick Smith, J.F. Marier, and Martin Beliveau will present Submerge in the Oligonucleotide Emerge, Best Practices for Developing These Complex Biologics. You can register for all webinars by visiting Certara.com. Yeah, can you go to the next slide, please? On behalf of Certara, I would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.